Bacterial transformation is the process of introducing new DNA into bacteria so that it expresses a new trait. In this example, students learn how gene regulation can turn on a jellyfish gene to produce green fluorescent protein in successfully transformed bacteria. This is a very similar process to how human insulin is made from bacteria for people with diabetes, and the basis for how we make genetically modified organisms, or GMOs. Students begin the lesson by watching and making some observations in their student guides about 2 short videos that feature organisms with green fluorescent proteins. Then, they'll learn about some existing crops, foods, medicines, and yes, burgers that have been genetically modified. Next, students begin exploring what a plasmid is and how, in this example, production of green fluorescent protein can be regulated by a specific transcription factor. We also introduce or review antibiotics' effects on bacteria and resistance conferred by plasmids. After reviewing experimental design for the lab and making some predictions, students will learn how to do bacterial transformation through reading, watching videos, and activities embedded in the student guide. Then we'll have students look at some representative results for the transformation, capture their observations, and identify some possible sources of error. In the final step of the lesson, we ask students to communicate what they have learned. Teachers may use a variety of assessments that work best for their classroom. We have included scaffolding for students to make a scientific model to help construct an explanation of the difference between transformed and untransformed bacteria. For students less familiar with constructing a model, we've included some instructions and examples here to help them get started. Please visit babec.org to find curricular resources for this lesson and to learn more about available lessons for your classroom.